Here. It dawns on me that I think the podcast finishes at 9 anyhow, so we need to get moving through this sort of thing. So during the first 6 weeks, we're going to cover more sort of practical aspects of organic chemistry. How do we conduct reactions? How do we separate molecules? And starting on Thursday, how do we identify what functional groups are present, how many carbon atoms there are in a molecule, what the carbon skeleton looks like, and so on and so forth. So today we're going to cover extraction and purification by chromatography, and then we'll do characterization later. And this is true of almost anything. It's true of a chemical reaction. So in the chart that we put up just before the break, right, you saw that if you take a compound with one functional group, you want to change that functional group into a different functional group, you react it with a reagent, and so innately at the end of the reaction process, you have a mixture. You have unused reagent, you have spent reagent that's been transformed into something else, you may have some of your starting material left, and you may have some of your product left. And so almost all chemical reactions don't just suddenly precipitate something nice and you've got pure product. Almost all cases, we need some form of separation of the chemical compounds that are there to obtain a pure product. And once we have a pure product, then the third step of all reactions is to identify it, to determine the structure, to see whether, or to prove essentially, whether the structure is the structure that you set out to make in most reactions. This is also true of isolating compounds from nature, such as Taxol that we saw at the very beginning of tonight's lecture. So Taxol is isolated from the yew tree. Yew tree probably contains tens of thousands of different compounds. An E. coli cell contains, it's estimated, about 5,000 different chemical structures, right, including all the proteins, nucleic acids, so on and so forth. There are around about 5,000 compounds in E. coli and around about 100,000 compounds in the human cell. So if you want to do metabolomics, which is a fashionable thing to do these days, to work out all the compounds that are present, all the metabolites that are present in a cell, you have a big separation problem. You have to separate out, ideally, all of those 100,000 compounds from a human cell, measure their quantities, and see how that metabolic profile, for example, changes as a function of disease or as a function of altered input, for example, energy supply. So separation is a huge problem, right? And so classically, most of separation science developed in chemistry. Now it has expanded into biochemistry, where we want to separate out proteins, nucleic acids, where we want to separate out different length stretches of DNA, for example. Separation is a problem all over science, and so it's important to understand some of the basic principles of separation science. It's called separation science nowadays because really the business of separating things is a whole science unto itself. Very hard to give this just treatment in one lecture, but we will try. So you in the lab will be required to perform separations. In experiment 1, you're given a very simple mixture, probably of 2 or 3 compounds, I forget now. Okay, it's a mixture, and now you tell your challenge is to then separate those compounds out, and you're going to use very simple identification technique. You're going to use the melting point, comparing the melting point of your purified material after you've separated it with a bunch of known melting points to determine the structure of, I think, a carboxylic acid. Okay, so that's interesting. Drugs and pharmaceuticals. Separation science here enormously important. So in the pharmaceutical industry, I believe it's around about 1 in every 5 pills that are manufactured in the pharmaceutical industry is broken open and analyzed through separation science to ensure that that particular batch is coming through consistently with the right dose, with the right quality, the right purity of pharmaceuticals. So there's a huge amount of separation science done in the pharmaceutical industry. And it requires that degree of stringency to ensure that you contiguously produce a safe product. So when you complain about the cost of your next prescription, bear in mind that a fifth of that cost has gone down the tubes to make sure that the product is safe to use. Where do drugs and pharmaceuticals come from? Many come from chemical reactions, so they're about half of all useful human pharmaceuticals are made by organic synthesis, by classical organic chemistry reactions. So where do the other half come from? The other half of the compounds, like Taxol, that come from natural sources like the yew tree. Penicillin, for example, from Penicillium species, small organic compound containing functional groups that you can identify. And this is produced by Penicillium. And again, here we again have a separation problem. In the pharmaceutical industry, penicillin is produced in 100,000-liter vats of fermented penicillium. It's been genetically engineered to produce the maximum yield of penicillin. But still, of course, what we have is a whole load of bug juice containing penicillin, and chemists have to go in there, sort the mess out that the bio people have made, okay, and purify that penicillin, analyze it, and so on and so forth. Okay. So 50%, or perhaps the slight majority, of pharmaceuticals sold today are a result of a lead isolated from nature. Aspirin is a classical case and is, of course, used to treat pain. In fact, people have recognized for a long time that a compound called salicylic acid that is present in the bark of yew trees is, in fact, the active ingredient that causes release of pain. So there's the yew tree and, anyhow, there's the tree, okay? And within all those compounds that are mixed in there is some salicylic acid. How could we get that out and purify it to use it as a drug? So there's the structure of salicylic acid. It has this aromatic ring, this benzene ring. Okay, there's one of the functional groups that we met. There's an OH group that looks like an alcohol. In fact, this is a special type of alcohol. It's a phenol when the OH is attached to a benzene ring. And in 2OB3, you'll look at those types of compounds and study their reactivity. It's also got a carboxylic acid functional group, CO2H, which is a separate little group of 4 atoms, the carbon, the 2 oxygens, and the hydrogen, essentially together form a functional group that means that the compound is acidic. So salicylic acid tastes very bitter because of the carboxylic acid functionality. So it was Bayer who recognized that you could do a chemical reaction on salicylic acid, a natural product, and convert it to a semi-synthetic drug. So semi-synthetic drugs are drugs where we start with something in nature, we do some chemistry to it, and this is really the third category. So we have totally synthetic, semi-synthetic, and direct natural products are the 3 types of drugs that occur. So in this reaction, we're going to take that alcohol and convert it to an ester functional group using acetic anhydride and acid. It's actually the formation of an ester is one of the reactions on the cheat sheet that we looked at before the break. And this makes acetyl salicylic acid. This is now less bitter. We've removed that phenol OH, acidic OH group, and protected it essentially, converted it to a different functionality, makes the compound less bitter, and so we can ingest aspirin. And then, in fact, in the body, the ester group is cleaved off by enzymes called esterases to regenerate the salicylic acid, which is the active compound. And Bayer got, of course, incredibly rich by figuring this little bit of chemistry out. Okay, so how do we get at chemical compounds? How do we isolate them? How do we purify them? And then how do we know what we have? And that's spectroscopy. So isolation and purification is what we're going to talk about today. So one of the basic sort of techniques when we have a mixture is to do extraction. Extraction is something that you will do in the 2O lab in a separatory funnel. Basically, we're going to pull out a compound out of one liquid phase into a different liquid phase. So we're going to have an organic phase that will dissolve organic compounds, and usually an aqueous phase that will dissolve water-soluble compounds. And so compounds will go into one layer or the other, and we have effectively got a very crude separation of compounds into two categories: the ones that are water-soluble and the ones that are organic-soluble. So this is rather like a cup of tea, right? We're used to making a cup of tea. We take solid tea leaves containing tannins and caffeine. We pour on boiling water, and the caffeine is removed from the leaves. The leaves are left behind. The caffeine dissolves, in this case, in the water phase. So quite often for natural product isolation, we grind up the source and allow it to stand in a solvent. So a solvent usually is an organic solvent. It's a chemical compound that contains a functional group, usually a very simple chemical compound that is cheap and commercially available. Some solvents might include hexane as an alkane, right? You can buy 4-liter bottles of hexane. So that would be a very non-polar solvent. Remember, just before the break, we saw that alkanes are very non-polar molecules. Or we might use something like methanol, an alcohol that is much more polar, a more polar solvent. But in either event, we grind up the source, allow it to stand in the solvent. So what solvent should we use? It depends on what we are trying to dissolve. In the case of the cup of tea, we want to dissolve caffeine. Caffeine, it turns out, is a polar molecule. It's got 4 separate nitrogen groups in it. They're basic. So caffeine is water-soluble. So when we do our extraction of tea, caffeine easily moves from the solid phase into the liquid. Polar compounds, so polar compounds like caffeine dissolve well, usually in polar solvents, and relatively poorly in non-polar solvents. And we'll look at this in more detail in Chapter 2, I believe, a little ways down the road. But it's the principle of like dissolves like. So we get better non-covalent interactions, we get better intermolecular forces when like molecules are dissolved in like solvents, and poor intermolecular forces when we try to dissolve a polar compound in a non-polar solvent. So non-polar compounds will dissolve in non-polar solvents. So here are some solvents. And this is often called the allotropic series. It's important in chromatography as well as simply choosing a solvent to do an extraction. Hexane, hydrocarbon compounds containing no oxygen or nitrogen functional groups are the least polar. Alkanes, aromatic compounds like benzene, that should be benzene, of course, are slightly more polar, because they have carbon-carbon double bonds, but still very little polarity in the bonds. Ethers, we saw just before the break, a bit more polar. So you have functional groups now in the molecules that have polar bonds, bonds between atoms of different electronegativity. In ether, it's the carbon and the oxygen that have different electronegativity. It gives rise to a polar bond, and we have a more polar molecule. Other common molecules, chlorinated solvents, like chloroform, CHCl3. Okay, so chloroform used to be a very popular solvent in the lab. In fact, that you're going to do, you will probably more likely use dichloromethane or dichloromethane, as it's written there. CH2Cl2, because chloroform, it turns out, well, for one thing, as you're probably mostly familiar, chloroform's an anesthetic, kind of knocks students out, right? So if you started not very with it in the beginning of the lab, by the time you've inhaled a lot of chloroform, this is not so good. But more seriously, chloroform is also a liver toxin, so we don't use that, and we would use something like dichloromethane. So these are alkyl halide-type functional groups. Esters, like ethyl acetate, a bit more polar. Okay, and then we talked before the break about how an alcohol is much more polar even than an ether, because we have all these polar carbon-oxygen and oxygen-hydrogen bonds, and then water is about the most polar solvent in the series. So the like-dissolve-like thing tells us that nonpolar fats and lipids, compounds that are very nonpolar, very fatty, dissolve in hexane, whereas very polar compounds such as salts, sodium chloride, of course, is not soluble in hexane, but soluble in water. So we have this series of increasing polarity solvents, compounds, organic compounds that are readily available, cheap, available in large quantities. And this is, in fact, the sequence that we would use to elute compounds off a chromatography column for the same reason, as we'll see. So in practice, compounds generally partition between a pair of non-miscible solvents. So if I mix water and dichloromethane, water and dichloromethane don't mix. They're too different in polarity to want to mix. So I would get a mixture containing two layers, a dichloromethane layer, which in fact is on the bottom. And a word to the wise when you do lab 1, remember which layer you are after at each point. So the lower layer will be the organic dichloromethane layer, and the upper layer in a mixture of dichloromethane and water would be the actually lighter water layer. So if I take a mixture of those two and I add a chemical compound, it will partition. Some of the molecules will dissolve in the dichloromethane, other molecules will dissolve in the water. And the number of molecules that we get in, for example, the dichloromethane layer depends on the polarity of the compound. So we can then we generally find that most compounds will prefer to be in one layer or the other. And so by doing multiple extractions, by doing this process several times, we can pull out the compound that we're looking for. So let's look at extracting a compound X from water into dichloromethane. We take a device called a separatory funnel. It has an odd shape, but the shape is designed to help you to separate the phases. Okay, and you'll experience this next week. So we have our separatory funnel, the device in the middle. We have an empty flask at the bottom, a stopper, and we pour a mixture of dichloromethane and water into the separatory funnel. We get two layers. We add the appropriate compound that we want, shake well, put the stopper in, shake well, and when you're shaking a separatory funnel, always make sure, as in B here, that you open the tap every couple of shakes. Dichloromethane is quite volatile. Pressure builds up inside the sep funnel, and you have to release that pressure. If you don't, then the stopper comes blowing out, and all your mixture goes everywhere. So you, as you're doing lab 1, okay, watch for that. Make sure that you vent the sep funnel. So then we stand the sep funnel back up, and now we have our desired compound in one of the two layers, the upper layer or the lower layer. We pour out the lower layer through the tap. We stop right at the interface. The separatory funnel is designed, you see it narrows towards the bottom. It's designed to allow you to just catch everything just before and everything just after the interface between the two layers. So we collect the two layers in two separate flasks, and consequently we have now achieved a separation of the compounds. The compounds that want to go in the water layer will be in the water layer. The compounds that want to go in the organic layer will be in the organic layer. So at the end of this process, we have a very crude separation. We've separated one class of compounds from another class of compounds. This simply points out that most compounds are not 100% soluble in one phase and 0% in the other phase. So in fact, we have a partition coefficient. It's actually an equilibrium constant. It's the concentration in solvent A divided by the concentration in solvent B. This is a little different. This is really not different from any equilibrium constant, but in level 1, used to equilibrium constants between different molecules that are reacting. Here, this is simply an equilibrium constant for the concentration in the one solvent divided by the other. So if we have a gram of compound that has K equals 9, and we put it into a separatory funnel with 100 mils of dichloromethane and 100 mils of water, then K equals 9, okay? And if you do the calculation from the equilibrium constant, the ratio of concentrations is 9 to 1. So you will have 0.9 grams of your compound in the solvent that it wants to be soluble in, and only 0.1 grams in the solvent that it doesn't want to be soluble in. So how do we get a better yield? How do we avoid losing the 0.1 grams? We do a second sequential extraction of the solvent that the compound is less soluble in. We extract again. The equilibrium establishes itself again. So after 2 sequential extractions in a separatory funnel, we have 99% of the compound in the desired phase, for the sake of argument, dichloromethane. So this illustrates an important point. When you're doing extractions in a separatory funnel, you need to do 2 or 3 repeated extractions in order to get a good yield. Good yield means high mark in the lab. High mark in the lab means high grade in the course. High grade in the course means go to med school, get rich, have a happy life. For the rest of you with a low yield, not so much. Okay. Right. Okay. So we've now managed to separate out, for example, our compound into the dichloromethane layer. Now we need to remove the dichloromethane and maybe even separate the compounds that were soluble in the dichloromethane layer even more. And there are some classic techniques for doing this, which apply only in a few particular circumstances. And then there's chromatography, which applies to almost any separation, further separation of compounds. So for volatile liquids, we can do a distillation. I think everybody's familiar with this. So if, for example, we had dichloromethane, a mixture of 2 compounds dissolved in dichloromethane, distillation, the first thing that's going to come off is the volatile dichloromethane. We can boil that off and leave the 2 compounds behind, and then we can raise the heat and distill some more, thereby separating the 2 compounds based on their boiling point. So here's a classic sort of simple distillation setup. You will actually use a somewhat different distillation setup in the 20A3 lab, but the principles are the same. We put our mixture in this flask on the left-hand side, we heat it, eventually one of the most volatile compound will boil, the vapor rises, it goes over the thermometer so that we can record the boiling point of that compound. The vapor then travels through a condenser and we collect the product on the right-hand side. And this, of course, distillation is a technique that's as old as the hills. People have been distilling ethanol for reasons that I couldn't begin to imagine out of fermentation products for centuries, if not millennia. Right? So this is all fairly familiar. Okay, if things get really difficult and the boiling points of our compounds are quite close, we can do fractional distillation. This is basically refining oil, essentially, is fractional distillation on the thousands of ton scale. We take the oil that contains many different organic compounds, some short-chain alkanes that are more volatile than the long-chain alkanes. We boil the whole lot, and we collect different fractions that distill at different temperatures. So fractional distillation is a slightly more sophisticated technique that allows you to get a better separation of volatile compounds. So distillation works well if we have volatile compounds, but what if we have non-volatile compounds? Then distillation, of course, is not going to work. And so most organic compounds, things like Taxol, we'd have to boil them at 500 degrees Celsius in order to get them to distill, and that, of course, destroys the compound. So distillation is limited because we need compounds with reasonable boiling points. So if we now move to solids, which we can't distill, we have an alternative classical technique called crystallization. So this is the simplest technique for purifying a solid. So we take our solid, we dissolve it in a suitable solvent, bearing in mind the like-likes-like rule. And so we would find a suitable solvent. We dissolve it in the solvent when it's hot. Okay, so we use the minimum amount of boiling solvent to dissolve the solid. We might filter if there's some insoluble junk in there. That removes the junk. But then we basically cool the solution. And so as we cool our saturated hot solution, as it cools down, the solubility of the compound decreases. And your chemical compound will form crystals, and that will give you purification because small traces of impurity will not be saturated, and they will remain in solution. So slow, gentle crystallization of an organic compound. You've all grown copper sulfate crystals or sodium chloride crystals, I expect, right? And this applies equally well to organic compounds. Slow cooling of a solution of an organic compound in a suitable solvent, and there is the trick. You have to find the suitable solvent. But that will give you purification so that we end up with purer material at the end of recrystallization than we had at the beginning. So we now have crystals. We will collect those on a device called a Buchner funnel. You'll figure out how to use that in the lab. And that's a way of simply removing the excess solvent and impurities. We dry to remove the residual solvent, and then we test for the purity of the compound. We can test for purity by recording the melting point. And if we know the literature melting point of organic compounds, we can compare our melting point with the literature, and we can begin to characterize the compound to determine its structure. That is all very classical techniques. So you might wonder, well, what happens if I can't find a suitable solvent? And that happens a lot with organic solids. What happens if I don't have a volatile compound? I can't distill, I can't crystallize. Oh, shit, what am I going to do? Okay? And so the answer, of course, is to use a technique that works irrespective of whether we have a liquid or a solid, whether we have a high or a low boiling point, whether we have a crystallization solvent or not, and that technique is chromatography. So chromatography is used in biology, it's used in chemistry, and basically all chromatography is the separation of components of a mixture based on the differences and the rates at which they are carried through a stationary phase by a mobile phase. So all forms of chromatography have a stationary phase. It might be a solid matrix like silica gel, that's what you're going to use in the lab, packed into a column. So you have a long column of silica gel, which is sitting stationary. We're going to have a mobile phase, and in the case of the chromatography that you're going to do, the mobile phase is going to be a liquid. It's in fact going to be one of the solvents that we have in our allotropic elution order. So we're going to have a liquid phase that's going to pass slowly through the column. And molecules of different compounds will absorb more or less onto the stationary phase, and they will partition between the stationary phase and the mobile phase. That word partition we've used already, right? We used partition when we talked about separation of things by extraction in the separatory funnel. We talked about how you could write an equilibrium constant for compounds in the organic phase versus in the aqueous phase. And here, partitioning in chromatography is exactly the same process. Molecules of a compound will partition between the solid phase, where they're bound to the solid and don't move as the mobile phase passes by, And then some of them will be out in the mobile phase. There'll be an equilibrium between mobile and stationary phase. Molecules will be exchanging between the two phases very rapidly, i.e. they are at equilibrium between the two phases. And so, depending on the nature of that equilibrium, if the molecules are 90% on the stationary phase, then they're only 10% in the mobile phase. Only 10% of the molecules are moving as the mobile phase moves forward, and the compound will stick quite tightly to the column. If, on the other hand, 90% of the molecules of your second compound, your impurity, for example, are in the mobile phase, and only 10% are stuck on the stationary phase, then that compound will travel rapidly through the column. There are many different types of chromatography, but all are based on the same principle. So we put the stationary phase in a column or on a planar surface, and this is thin layer chromatography, where you're going to take a sheet of aluminium foil or plastic onto which has been put a thin, even layer of silica gel that we're going to use for separation. So the solid phase may be in a column or on a planar surface. Usually it's a solid, and common solids include silica gel that you're going to use in the lab. It's the commonest reagent, stationary phase for organic chemistry. Alumina, less common, aluminium oxide. But there are a whole range of stationary phases for biological chromatography. There are ion exchange resins, size exclusion gels, whole range of stationary phases, but they're all the same. They're all compounds that are packed into a column or onto a plate that do not move when a solvent travels through them. The stationary phase can even be a liquid. In gas-liquid chromatography, which you may do in one of the experiments, then our mobile phase is a gas, and it's actually traveling past a stationary phase, which is a liquid. How do we make a gas pass over a liquid? The liquid is actually coated onto the surface of solid particles, and the exchange process in GLC, gas-liquid chromatography, involves molecules partitioning between the gas phase, which is mobile and traveling, and the liquid phase, which is on the stationary particles, which is not moving. So the mobile phase moves over or through the stationary phase, carrying with it the analyte mixture, and the mobile phase can, of course, be a gas or a liquid. So in gas-liquid chromatography, the mobile phase is a gas, and the stationary phase is a liquid. But in other forms of chromatography, silica alumina, where the stationary phase is a solid, then the mobile phase will be a liquid, a solvent, usually from our list of solvents. So a solute is distributed between the mobile and stationary phases. We can write an equilibrium there, and we can define what's called a distribution constant, but it's another equilibrium constant, K, which is the concentration in the stationary divided by the concentration of compound in the mobile. So different compounds will partition differently between the stationary phase and the mobile phase. So they will have different equilibrium constants. They will travel at different rates through the column or up the planar surface. So in an ideal world, every compound that we want to separate will have a unique affinity, a unique equilibrium constant, distribution constant, a unique affinity for the mobile phase. And if that is the case, then we can separate everything in our mixture. So because different compounds are bound more tightly or less tightly to the stationary phase, they will be more or less in the mobile phase. They will travel faster or slower down the column or up the plate and can be separated. So in column chromatography, you generally have a stationary phase like silica gel, and the mobile phase is a solvent. And here now it's a question of choice of solvent. We need a solvent that will dissolve the compound of interest. So remember that like dissolves like. So in choosing a mobile phase, we don't want a mobile phase in which our compound is insoluble. So we have to have something in which the compound is soluble, but we also need a reasonable polarity so that the compound can partition to a reasonable degree into the stationary phase. But if we choose a compound with too much polarity, all the compounds will remain in the polar solvent and come off the column. If we choose a compound with too little polarity, everything will stick to the column. So here is a column. We have a packed column of silica gel. We begin by putting solvent in there, and once the column is completely wet, we load on our sample as a nice narrow band at the top of the column. Here, this is illustrated by two compounds, A and B, being separated. We then start adding mobile phase, and we keep running mobile phase through the column. And A and B move as bands down the column. In this particular case, A is moving slightly faster than B, meaning that A has a slight preference over B for being in the mobile phase. Molecules of A are better solvated in the mobile phase than molecules of B. B then, if you like, another way of thinking of it is that B has more of a preference to bind to the stationary phase. And in silica gel chromatography, silica gel is very polar. Silica gel is always the most polar species in the mixture. So if I have a non-polar solvent or a less polar solvent and a more polar stationary phase, then like dissolves like, and consequently the polar molecules will bind to the silica. So the elution order is always that the more polar compound comes off last on silica gel. You have to think about this a bit. Everybody thinks like dissolves like. Therefore, something like an alkane, right, would, in a polar solvent, would bind to the silica. But it's not true because the silica is more polar than any solvent you can choose. So alkane molecules never want to bind to the silica. They will elute first through the column. They'll stay in the mobile phase because we never want to put a non-polar alkane bound to the silica. Ether, a bit more polar than an alkane. So an ether has a bit more polarity, a bit more preference to bind to the silica gel, which is very polar. And so an ether will elute second. It will travel more slowly down the column than an alkane. So A here could be an alkane and B could be an ether. Alcohols, as we saw before the break, even more polar. So alcohols will bind more tightly to the stationary silica phase because it's very polar. Alcohols will elute third. So if we had a column here, A, B, C, it would be alkane, A, B would be the ether, and C would be the alcohol traveling even more slowly through the column. As we work down, the bands gradually separate more. A and B now in the third figure are now becoming fully separated. And eventually we can have pure A coming off the bottom of the column, and then in a separate flask we can collect pure compound B. If I was actually to put a detector on the end of the column, my detector signal would be 0, and then I would get a peak coming off the column corresponding to molecules of A. That's the less polar compound, followed by a second peak from the more polar compound B. And of course, if I had an alcohol, then it would come off third. We would have a third peak. So what we've done is use column chromatography to separate compounds according to polarity, with the least polar compound coming off first and the more polar compound coming off last. Now, if I used a hydrocarbon here as my solvent, if I used hexane, my alkane would come through the column, but the ether and the alcohol would never come off. My My solvent is very nonpolar. The ethers and alcohols don't want to go in there. They're more polar molecules, so they will bind to the silica, not to the solvent. So how do we deal with that? How do we get the very polar compounds to come off this column? And the trick then is to increase the polarity of the solvent. So you can go through the allotropic series, the series of solvents that we saw earlier on, eluting one after another. Okay, and gradually, as we change from hexane, we will get the hydrocarbons coming off in hexane. As we go to an ether solvent, more polar compounds like ethers will come off. As we move to a very polar alcohol solvent, very polar compounds will come off the column. Okay. So retention time is a measure of the time that a compound takes to pass through the column, doesn't matter. And solvent and stationary phase must be chosen and matched to the polarity of the compounds to be separated. Yeah.
Sorry, in the graph that you showed before,
The detector could be detecting just about anything. It might be UV absorption, ultraviolet absorption, right? So as the molecules come through, they absorb UV light. But that is a topic to be dealt with in 2OB3. Okay? Or you could have a mass spectrometer hooked up to the thing, okay, analyzing the molecular weight of the compounds that are coming off. And that's a topic that we will look at in a couple of weeks' time, next week. Okay? So just an imaginary detector that somehow detects molecules coming off. If your compounds are colored, the detector is your eye. You can actually see the bands of different color traveling down the column. And so then you can just use visual detection. Okay, so if the affinity for the mobile phase is too great, the compound passes through the column with the solvent front and is not retained. If the affinity is too great, the compound retained on top of the column does not move. So we're aiming for an intermediate point. And here is our allotropic order again. Okay, slightly different solvents in this list. But choice of solvent polarity is the key to obtaining a good separation. One of the reasons that chromatography is so efficient and so good is that you don't have to just stick to one solvent. You can use a mixture. You can use 10% ethyl acetate in hexane. There's a very common one, mixture of ethyl acetate and hexane, for non-polar compounds. You can use different proportions of methanol and chloroform for polar compounds. You can choose any solvent you like. Notice the difference. Distillation required us to have volatile compounds that would not decompose when we heat them. Recrystallization required a solvent in which my molecules were soluble when hot, but relatively insoluble when cold. Okay? Here, all those little requirements are removed in chromatography. All we need is to be able to pick a solvent all the way through from hexane, very nonpolar, to neat methanol, which of course is very polar, or all points in between. There's an infinite number of permutations here to choose a solvent. And so that enables you, with care, to achieve very tricky separations. Yeah. Sorry?
Why is it actually called chromatography?
Chromatography comes from the Greek chromos, meaning color. And in fact, it originated as the separation of colored compounds. Okay? So that's what we were just saying, that you could use your eye as a detector to see the different bands coming down the column. Okay. So this just says what we said just a minute ago. For chromatography on silica gel, both for column and TLC, increasing solvent polarity makes all compounds travel faster. That is counterintuitive. I know everybody every year struggles with this idea. But the more polar the solvent, remember that there's three different interactions here, right? There's solvent-molecule interactions, there's molecule-silica gel interactions, but the third one that's also at play here is the solvent-to-silica interactions. Okay? And so we have to balance these. So in effect, going to a higher polarity solvent is rather like displacing the compound off the silica gel. So more polar solvent binds more tightly to the silica gel and displaces molecules of your bound substrate. Okay? And so all compounds travel faster, and we begin to elute increasingly polar molecules that otherwise, with a non-polar solvent, stick to the column. So an alkane elutes in any solvent. An ether does not elute in hexane, but will elute, for example, in ethyl acetate. An alcohol requires ether to elute it. And as our polarity goes up and up, a diol, a compound with two hydroxy groups, now requires, might require methanol chlorophyll mixture in order to get it to elute. So the diol with two OH groups, two functional groups, both of which are polar and bind to the silica gel. Now we need a very polar solvent to push it off the silica gel to replace the molecules absorbed on the silica gel by solvent molecules. Any questions? Yeah. You mean like sort of experimentally? Well, basically, if it comes off the column, it's displaced the compound, right? Yeah? Yes? Okay? All right. So that's that. So let's suppose that we've done a chromatography column, and we've separated compounds. We've now got A and B and C. Our three compounds are separated in three separate flasks. How do we know, how do we identify what is present in the different fractions that I have collected from the column? And one of the best ways to do that, or to determine what's in which fraction, is thin layer chromatography. So TLC is excellent for analysis of mixtures. The absorbent, usually silica, is supported on a thin coating, or is supported as a thin coating on a flat surface, usually glass, aluminum, or plastic. Okay? So we have a plate of aluminum, thick aluminum foil, right? And onto that is about a 0.1 millimeter thick layer of silica gel, and that is our stationary phase. We do something called spotting the compound on the plate. So we put a little spot of my compound onto the TLC plate, and you'll do this next week. And then we place the plate into a jar containing solvent. And solvent travels up the plate via capillary action. So here's a picture of that. Okay, we cut a piece of TLC plate. This will be given to you in the lab, right? We make with pencil, because ink has got organic compounds in it, and we'll make a nice, pretty elution picture all the way up the plate. So use a pencil to mark a spot. We just put a tiny amount of sample, roughly speaking, less than a microgram, on the order of nanograms of compound on here. So the one mistake that most students make in doing TLC the first time is that they think more better, right? And put a gallon of compound on there. So we're aiming for maybe 100 nanograms of compound. So we can take a dilute solution of your compound in a solvent, spot it on the plate, and you really need very, very little. So remember here that the sample must be spotted up the plate enough that it doesn't sit below the level of the solvent in the tank. So we arrange everything all this way. The solvent ascends up the plate. And now we get exactly the same partitioning that we had in the chromatography column. We have the baseline, molecules travel up the plate. Molecules that bind to the silica tightly don't travel very far. They're predominantly in the stationary phase on the silica on the plate. Molecules that prefer to be in the solvent, non-polar molecules, will be in the solvent. They travel forward with the solvent front, and so nonpolar molecules travel further on a TLC plate. Polar molecules travel less distance. So we end up with a series of spots on the TLC plate, and the distance traveled determines the nature of the compound. So the nature of the compound determines the distance traveled. We have something called the RF. RF is the ratio B to A. It's the distance traveled by the compound divided by the distance traveled by the solvent front. So you should measure the RF of your spots in experiment 1. And that RF is a constant for a particular compound in a particular solvent on a particular TLC plate. So if I had a second plate, if I had an unknown compound on here, and my component spot was a known compound, let's suppose that these were two analgesic drugs, and I want to know are they the same or different. I spot them both on the plate. The solvent travels the same distance up the plate. On the left-hand side, moving one spot forward. On the right-hand side, moving the other spot forward. If both spots move to the same distance, are they the same compound? Yeah. It's not bad evidence. Would you go to court with that if you wanted to prove that it was cocaine? No. I don't think any judge would accept the fact that one TLC plate with two compounds traveling the same distance proves that they're the same molecule. But it is some good evidence. And as we'll see on Thursday, all of spectroscopy is about building bits of evidence rather than having one simple proof. So ideally, each component has a different RF and travels a different distance up the plate. And we can then visualize those spots by a variety of techniques. One that you will use in the lab is to shine a UV light on the plate, and you will see the individual spots light up. Really, if this sounds complicated, okay, don't worry. When you do this in the lab, it will all seem clear to you. There are many other types of chromatography, including preparative layer chromatography. That's where we use a thicker layer to do preparative separations. HPLC is probably something that's familiar to many of you. High-performance liquid chromatography, also called high-price liquid chromatography because it's a lot more expensive and we need a special machine. Gas chromatography, all these techniques do the same thing. They separate molecules based on how fast they travel through a stationary phase on a mobile phase. Other types of stationary phase, reverse phase silica. Now we can separate based on hydrophobicity as opposed to molecules being separated on polarity. Paper chromatography, used to separate amino acids in biochemistry. Chiral phases that separate enantiomers, something that we'll talk about later in the course. Ion exchange resins, size exclusion, many, many more different stationary phases. So chromatography is hugely versatile. We can change the stationary phase through a wide range of different possible matrices, and we have almost infinite control on the mobile phase.